Then you end up at something called the nephron loop. So then the first part of nephron loop is the descending limb, and then you have the ascending limb. So this is what we call the loop of Henle, or your book calls it the nephron loop. Same thing. All right, so this is what we have. So we have something called a countercurrent multiplier. So I know it sounds like math, but it does involve numbers, but it's not multiplication what you think it is. But the thing is that countercurrent. So what's happening here? Well, notice that the descending limb is going down into the renal medulla, and then up and ascending out of the medulla into the cortex. So this is the countercurrent. Notice that these currents are going in opposite directions. But what's happening here? Notice that different things are moving in the descending limb versus the ascending limb. So another figure in your book, I think, that illustrates this a little better is this figure. So all these black dots, these are solutes. In particular, this solute we're primarily concerned with is sodium chloride. So what we have here is the filtrate. Remember, things, small ions do, and water move out from the glomerulus and filter through into the capsule. But what happens here? So this is where osmosis comes back with a vengeance. So remember, if you have a very salty solution and a more dilute solution, which way does the water absorb in osmosis? Like you have say 500 moles of solute and 100 moles of solute on the other side. Which way does the water go? It goes to the saltier side, right? So remember, the saltier, the more sugar, the more protein a fluid has, the more it's going to attract water in osmosis. So think about the renal medulla. It's actually got it's very, very hypertonic compared to the inside of the renal tubule. It has a lot of sodium chloride and it has a lot of urea. Even though we think of urea typically as a waste byproduct, it also has the osmolarity. It also attracts water as well. So you have the super, super salty, super concentrated urea concentration, and this is going to move water out. So what we see here is that this descending limb is impermeable to solutes. So again, this is a semi-permeable membrane. So things like sodium chloride aren't going to move through, but water can move through this limb. So what they don't show in this picture is that there should be actually way, way, way more salt and urea concentration. So you actually have a very salty solution, not so salty here. So as you go from here down to here, you're going to draw out more and more water. And what happens? What do you see with the salt concentration? As you draw out more water, the salt concentration goes up. So it's kind of like if you're cooking at home and you say you have a soup stock. What happens if you leave the soup boiling for an extended period of time? Well, the soup loses water, and what happens to the concentration of that flavor inside the soup? It gets more and more concentrated and stronger, right? Same thing is happening here. As you're losing water from the tubular fluid, it's concentrating the salt over here. So once you're finally in the renal medulla, you have this really salty solution inside the renal tubule. Now the salty solution is going to move up in the ascending limb. Now something has changed. You no longer have osmosis. Why? Because now this is what we call also the thick part of the ascending limb. So this thick tubule wall is actually not only impermeable to solutes in terms of passive diffusion, they're impermeable to water as well. So they're not going to allow water to osmose. Osmosis has stopped at this point. But what we have here are these proteins. These are co-transporters that are moving both both sodium and chloride from the inside of the tubule to the surrounding area. So what's happening here? This is why you start seeing the salt concentration start to drop in the tubule. But this is the cool thing. As you're moving salt into the surrounding area, this makes the renal medulla saltier. As you make the renal medulla saltier, this causes more water to be absorbed. So the thing is, it kind of amplifies itself. As you move water out due to osmosis, you have more salt. As you have all this salt and pump more salt into the renal medulla, you draw out more water, making this more concentrated salt, more salt to pump into surroundings. So this is why we call it the countercurrent multiplier. Why? by just again, the water is concentrating the salt is helping to draw out water. It's kind of like a positive feedback loop. It's kind of building upon itself and then exaggerating the both processes. So both processes are helping each other. Okay, so this is a nephron. Again, this is a key thing. The renal medulla is very, very salty. This is why the countercurrent multiplier works. Why? If it wasn't very salty, it wouldn't have the osmolarity to draw out water from the descending limb, or actually, yes, descending limb. And it would, and if it, again, you have all the sodium chloride being pumped out from the ascending limb, so you have a lot of sodium chloride, but also a lot of urea. And this high salt concentration, this is what helps to reabsorb water from the filtrate and back into the body. Now, this is the other thing. I only show the nephron right here, but the thing, in reality, this is how the nephron looks. So we did talk about PCT, now we're at the nephron loop. But look at the blood vessels. You do have the glomerulus, you do have the efferent arterial, but notice that all these blood vessels are right along the nephron loop. So this is a common misconception. Even though we're moving water out of the descending limb and we're also moving salt as well, it's not just going into the kidneys. Why? Because if you didn't have the blood vessels to carry away that excess water and salt, your kidneys would just accumulate water, they would accumulate salt, and there'd be huge saline bags full of water and salt. The thing is that this, again, you're moving water out of the filtrate, but you're also moving it back into your circulation. This is why it's very important to have these blood vessels not only in the glomerulus, but also reclaiming things you're trying to reabsorb. It just doesn't stay in the medulla, it actually gets reabsorbed into blood vessels. So you have these peritubular capillaries that are around the PCT and DCT. They are not the same thing as the vasa recta. The vasa recta refers to the capillaries that are around the loop of Henle. So I think actually it was it. I think it was Crash Course, and I think they actually said like these are the same, and like they're not the same. Peritubular capillaries are around the PCT and DCT, whereas the vasa recta, this is the one reclaiming all the water and all the salt that you're reabsorbing from the loop of Henle. Okay, and this is the other thing too. So another common misconception is that the efferent arterial. So here we have efferent flow, but here's our efferent arterial leading away from the glomerulus. It actually just doesn't go back into the renal vein. It actually loops back and collects all that salt that's being pumped out or reabsorbed from the renal tubule, and then it loops back down here to pick up all this water as well. So all this water is now being picked up, and now it can finally go back into the renal vein. Now it can finally go back into systemic circulation. So again, even though, again, why do we have those very simplified diagrams? Because if you've seen from the previous picture that the renal tubule and again, all the peritubular capillaries and vasa recta, this is what it looks like. It looks like a big tangled mess of balls and like tubes. So again, the nephron is complicated, but the end game you want, or what you really want from this is not only to get rid of your waste, but also prevent the loss of water, loss of ions, loss of solutes, loss of vitamins, loss of nutrients. So the thing is that it's very important to move it back into the blood. Okay, so filtration, reabsorption, and secretion. Again, the renal corpuscle is primarily refiltration, whereas reabsorption, you have the proximal convoluted tubule, and so the PCT does reabsorption and secretion. Loop of Henle. Main purpose of the loop of Henle is reabsorption of sodium chloride and water. Again, that countercurrent multiplier. By extracting water, you help to concentrate sodium chloride. By having all the sodium chloride and reabsorbing it, you also help to move water as well back into the surrounding tissues and back into circulation. Phew. Okay, yeah, that's the loop of Henle. Very complicated, but very important. Otherwise, every time you urinate, you would lose a lot of water, you would lose a lot of salt.